really great to be here, so I'll start. So I wanted to begin with the central idea that chronic inflammation systemically, but especially in the kidney, may lead to changes that mediate hypertension. So it all starts in the spleen, where T cell activation within the spleen results in homing to kidneys as well as vasculature. And this leads to chronic inflammation in the form of peripheral cytokines, which may then mediate kidney as well as vascular injury and lead to shifts in blood pressure. And so when you think of chronic inflammation, what comes to mind are regulatory mechanisms in place that govern this process. And one of the most dynamic ones that come to mind for me is neuroimmune mechanisms. So just interactions between the nervous system and the immune system. So we know that there's crosstalk between these two systems, wherein the immune system, by releasing peripheral cytokines that communicate with the nervous system, can prompt the secretion of hormones or neurotransmitters that in turn attenuate or augment immune system activity. And one example of this is the cholinergic anti-inflammatory pathway, which is an endogenous vagus nerve to spleen mechanism. And so the way that this pathway works is that the efferent vagus nerve synapses at the celiac ganglion, which causes transmission by the splenic nerve, and thus the release of norepinephrine in the spleen, which then binds upon beta-2 adrenergic receptors on T cells, thus prompting the synthesis and the release of acetylcholine into the spleen, which may then act upon alpha-7 nicotinic acetylcholine receptors on immune cells. And so basically there's secretion of pro-inflammatory cytokines by these immune cells, but others have shown that stimulation of the vagus nerve at the level of the efferent vagus is able to attenuate pro-inflammatory cytokine release. And so something that's really important to consider is that splenocytes may be principal in regulating this downstream anti-inflammatory effect. And so in this recent study, mice that were pre-treated with vagus nerve stimulation were protected from induced ischemia reperfusion injury. And interestingly enough, when these splenocytes were isolated and adoptively transferred to naive mice, these mice were also protected from renal injury with ischemia reperfusion injury. And so with that, our overall hypothesis is that impairment of neuroimmune mechanisms such as this cholinergic anti-inflammatory pathway may promote chronic inflammation and lead to changes that mediate hypertension and renal injury. And so to study this process, we use the disease model systemic lupus erythematosus, which is an autoimmune disease that primarily affects reproductive age women, and inflammatory kidney disease and cardiovascular complications are the most common causes of mortality. Also, there's dysautonomia in the form of decreased vagal tone in these lupus patients. And while we don't know exactly what causes lupus, there's interplay between genes as well as the environment. And this leads to abnormal immune responses wherein self-antigens activate T cells, which then prompt B cells to secrete autoantibodies and immune complexes that can deposit in all organ systems. And the mouse model that we use, it's been around for the past 70 years, and it's formed from the first generation cross between the New Zealand black and the New Zealand white mice, which we use as controls. And the female mice of the strain exhibit a lot of the same characteristics that you see in human lupus, such as the prognostic anti-double-stranded DNA autoantibodies, which are elevated compared to controls, renal injury in the form of albuminuria, and heightened renal inflammation in the form of renal TNF-alpha. And most importantly, these mice become hypertensive at around 34 weeks of age, as we and others have shown. So with this in mind, we ask the question whether the cholinergic anti-inflammatory pathway contributes to chronic inflammation as well as hypertension in our lupus mice. And in order to study this, we use two different pharmacological stimulators of the vagus nerve. And the first is CNI-1493, an experimental compound, and the second is galantamine, which is a reversible acetylcholinesterase inhibitor. So in the literature, others have shown that CNI-1493 increases efferent vagus nerve firing, and so here is direct IV infusion of CNI-1493, which increases amplitude. And CNI-1493 also dose-dependently decreases systemic inflammation in the form of TNF-alpha. Galantamine is also able to increase efferent vagus nerve firing, here seen in raising the heart rate variability in the high-frequency power domain. And like CNI, it also decreases systemic inflammation. So we used two sets of experiments, and at weeks 29 and 30, we measured renal injury in our lupus mice using albumin dipsticks and excluded mice with renal injury. we treated mice with CNI 1493 as well as galantamine, and at week 34, we implanted carotid catheters and measured mean arterial blood pressure. And later on, in a different subset of experiments, we actually isolated splenocytes for further studies. So one of the first parameters that we examined was the effect of chronic vagus nerve stimulation upon autoantibodies, which determine lupus disease severity. And so what we found was that that in our lupus mice, double-stranded DNA autoantibodies were elevated in both sets of animals, and treatment with CNI 1493 as well as galantamine was able to attenuate plasma concentrations, suggesting that these stimulators were able to decrease the severity of lupus over time. Also, galantamine was able to decrease the concentration of plasma anti-nuclear antibodies. Secondly, we examined renal inflammation and injury, and what we found was that in our lupus mice, there was elevated cortical and medullary TNF-alpha in our lupus mice, and treatment with both of these agents was able to attenuate expression of this pro-inflammatory cytokine. Secondly, in our lupus mice, there was a heightened albumin excretion rate, and CNI 1493 treatment attenuated the progression of albuminuria in our lupus mice. And so lastly, we examined the effects of chronic vagus nerve stimulation on blood pressure. And so what we found is that, as others have shown, and as we have shown in the past, lupus mice are hypertensive compared to our control mice. And while we didn't reach significance, there's a suggestive trend where CNI 1493 and galantamine were able to partially blunt or attenuate the rise in blood pressure that you typically see in these mice. And so future studies, we may employ telemetry, and perhaps it's just that the frequency or maybe the dosing or the total treatment duration wasn't long enough or early enough, but we will investigate this in the future. And so this brings me back to splenocytes, which I mentioned earlier. And so one of the more important questions that we wanted to investigate was whether chronic vagus nerve stimulation is able to condition splenocytes. And so this brings you back to this part of the cholinergic anti-inflammatory pathway, where the splenic nerve releases norepinephrine, which then binds upon beta-2 adrenergic receptors on T cells and therefore mediates the anti-inflammatory response. So in the splenocytes that we collected from our animals that we had treated with CNI and galantamine, we had plated them, and we expected pro-inflammatory cytokine release in the media. And so we hypothesized that by administering norepinephrine into the media that we could attenuate this pro-inflammatory cytokine release and measure any further reduction in this cytokine release. And so what we found in this preliminary data is that our splenocytes collected from lupus mice secreted elevated concentrations of TNF-alpha, and that CNI and galantamine treatment were able to further attenuate the release by splenocytes when we incubated all these splenocytes with norepinephrine. Likewise, we also found that our splenocytes from lupus mice secreted elevated IL-6, and we see a similar trend where CNI and galantamine both attenuate, both heighten the suppression or decrease the secretion of this pro-inflammatory cytokine. So, in fact, we have 4 major findings from this set of studies. The first is that treatment with these 2 different pharmacologic efferent vagus nerve stimulators was able to lower renal inflammation, protect from the development of renal injury, blunt the rises in blood pressure that you typically see in this murine disease model, and lastly, bolstered the immunosuppressive effects of norepinephrine when we took these splenocytes and incubated them in vivo. And so with that in mind, our working hypothesis is firstly that in lupus there's decreased efferent vagus nerve firing, which leads to a hypoactive cholinergic anti-inflammatory pathway in our mice. And this leads to heightened activation and homing of splenic T cells to the kidneys, which results in chronic release of splenic pro-inflammatory cytokines and further downstream renal injury, and a rightward shift in the pressure natriuresis curve and hypertension. And so from a clinical perspective, what excites me is that electrical vagus nerve stimulation has been indicated for the past decade in a variety of disease conditions such as epilepsy or treatment-resistant depression, and perhaps could also be indicated for autoimmune diseases, including SLE. And lastly, that vagus nerve stimulation's effects may be mediated through priming of these splenic lymphocytes. And others have investigated the role of the cholinergic anti-inflammatory pathway in other models of hypertension, such as angiotensin II mediated or SHR rats, but more studies are always great. So with that, I'd like to thank the Mat Lab, especially my mentor, Dr. Keisa Mathis, and members of the lab, of our small lab, and former members that I've had the pleasure to work with over time. And with that, I'll take questions.
So Dr. Abood has to have a question here, I think. No?
No.
No, why not?
Come on.
Right now, you plotted the vagus nerve supplying the celiac ganglion. You need to rethink that, and Dr. Tracy also should rethink that. But what has been shown is that the activation of the splenic nerve releases norepinephrine, which then induces from these cholinergic T cells that he had defined in the spleen, releases acetylcholine.
Right.
And it's the acetylcholine that is released from the splenic nerve sympathetic activation of the spleen that is supposed to induce the anti-inflammatory effect. So that's how.
Yeah, I'm aware of the.
But vagal stimulation is indeed anti-inflammatory, but I wonder whether that vagal stimulation effect is predominantly on the immune system in the gut, which is densely supplied by vagus nerve, rather than the spleen.
Right. I've been reading more into that, the vagal splenic anti-inflammatory pathway, and I do have to acknowledge that others have shown that there isn't a direct anatomical connection between the vagus nerve and the splenic nerve, but I do believe that there's at least a functional connection, but I will look into more papers about the splenic anti-inflammatory reflex.
How about lymph nodes and other secondary lymphoid organs? Can they be innervated by the vagus nerve?
That's something that we're really interested in, but.
And can you take these drugs for a long time instead of stimulating the nerve? Could you just give the animals these agents for years on end, and are humans these agents?
Well, interestingly, with CNI 1493, that actually underwent phase 3 clinical trials for Crohn's disease, but mostly because of its direct anti-inflammatory effects. And galantamine is used to treat Alzheimer's disease, so that can be safely tolerated for years.
This question.
I'm from Jackson, Mississippi, and I have a quick question. So you indicated that some of these animals, the SLE animals, don't develop kidney injury.
Right.
Have you looked at whether or not those that don't develop kidney injury have higher vagal tone?
That's a good question. We haven't really looked into that. We just got telemetry, which really excites me. So in the future, we'll be able to start examining heart rate variability. And yeah, that's something I've been interested in as well.
Thanks.
Okay, well, if there's no further questions, we'll bring our session to a close, and I would